Todd Dunn here. This is yet another video in my chronic lymphocytic leukemia series. Today I'm going to be talking about, can the current treatment paradigm produce a cure for CLL? What I'm going to be presenting today are my own personal thoughts based on my reading of the literature. These are not conclusions that I have seen in the literature or in videos by physicians and researchers. Again, these are my personal thoughts and should be taken as such. So, let's get started. What is a cure for CLL? Well, CLL, as you know, is a lymphoproliferative blood cancer which involves an accumulation of monoclonal mutated B lymphocytes. The current treatment paradigm in almost every case, with the exception of stem cell transplantation, addresses CLL by trying to eradicate the monoclonal cancerous B lymphocytes from the body. This is done in a number of ways. Traditionally, it was done by chemotherapy, in which the chemotherapy drugs interfere with cell proliferation, causing the death of the cells. More recently, monoclonal antibodies were developed, which attach to particular proteins on the surface of CLL cells, thereby labeling them for destruction by the body's own immune system. In this case, I'm talking about monoclonal antibodies like rituximab or obinutuzumab, which target the CD20 protein cluster on CLL cells or B cells. And there are targeted therapies which interfere with either the B cell receptor communication pathways by interrupting the pathways at either the BTK tyrosine kinase or the PI3K kinase. I'm talking here about drugs like ibrutinib or idelalisib respectively. Or they interfere with the BCL2 pathway, thereby triggering apoptosis in the cells or cell death. This paradigm of treatment is based on the idea that if you can get rid of the CLL B cells in the body, that you will eradicate the disease. Now, let's take a look at what this is based on. In the literature, if you read about the origin of CLL monoclonal B cells, you'll find that there are basically two proposed origins. The older proposed origin is that CLL B cells arise by mutation of an existing B cell, and then itself proliferates, producing CLL B cells. In this case, if the origin is an existing B cell, and then once the CLL cells are formed, they in turn proliferate, causing them to accumulate in the bloodstream, the lymphatic system, etc., then eradicating CLL B cells and also other B cells from the body should eliminate the disease. And in fact, the FCR treatment has produced some very, very long remissions that are still going on, approaching 16 or 17 years in some cases. And there are physicians who will suggest that this may well be a cure, that CLL B cells have been eradicated for those people and that they are cured. However, this represents only a small proportion of the people who had the FCR treatment, and others have relapsed, even though they too achieved minimal residual disease negativity, which means simply that given current technology, no CLL B cells can be detected in blood, lymphatic tissue samples, or bone marrow samples. That doesn't mean that they're eradicated, and the people who I've read on this topic say that the relapse occurs because even though we can't detect any CLL B cells, there are still a few left, and they will propagate and cause relapse of the disease. Now, there are also other studies which suggest that CLL B cells arise not only by a mutation of an existing B cell, which then propagates, but by mutation of stem cells and other progenitor cells in the bone marrow, and that those cells directly produce the monoclonal mutated CLL B cells from the very beginning, and that those cells then move out into the bloodstream and into the lymphatic system, where they propagate and cause the disease to progress. Now, these are two rather different ideas about where CLL B cells arise. As I said earlier, if they arise through mutation of an existing B cell, then eliminating B cells from the system should produce a cure. On the other hand, if the CLL B cells, at least some of them, arise initially in the bone marrow from stem cells and other more specific lymphocytic progenitor cells, then eradicating the B cell population, the CLL B cell population in the body, will not get rid of the disease, because the stem cells, which are producing the mutated cells, will still be there and will simply continue to produce those cells. And depending on their abundance, the disease will come back either quickly or very slowly. This points out, at least in my view, the idea that perhaps if the latter case, where stem cells are an ultimate source of CLL B cells, the idea that the current treatment paradigm, which targets B cells, really is not going to cure the disease. You may get some apparent cures, particularly for those people who have a particularly indolent form of the disease, and that maybe took 30 or 40 years to develop to the point where it became CLL. So if a person who's 60 years old and has that form of the disease, and you eradicate their B cell population, but not their stem cells that are producing mutant B cells, it might take another 30 or 40 years for them to develop CLL again. But they will develop it if they stay around long enough. But that person may appear to be cured even though they're not. So it comes down to, where are CLL B cells initially formed? And I'm of the opinion, based on my reading of the literature, that we're probably looking at a combination, that some CLL B cells, in some cases, perhaps dependent on the genetic phenotype of the cell, are formed in the bone marrow from stem cells and other progenitor cells. And perhaps in the case of other phenotypes, they are formed by mutation of an existing B cell, which then proliferates and forms the mutant CLL B cells. But what this points out is that if stem cells are a source of CLL B cells, then a treatment which targets the mutant stem cells will be required to cure the disease. You can't simply eliminate the B cell population, you also have to eliminate the mutant stem cell population, which means that research would need to be directed at characterizing the mutant stem cells so that they can be specifically targeted, rather than what is done in the case of stem cell transplantation or bone marrow transplantation, in which intense chemotherapy is used to wipe out not only the B cell population in the body, but also to wipe out the stem cell population in the bone marrow, which is then replaced with donor stem cells, which produce normal B cells. Now, if bone marrow transplantation didn't have such a high, unacceptable mortality rate, it would be a nice cure for CLL. Unfortunately, it doesn't always work out that way. About 30% of people who have bone marrow transplants die during the procedure. Another 30 to 40% have relapse even after they've survived the bone marrow transplantation, perhaps because the chemotherapy didn't completely eliminate the B cell population or the stem cell population in the bone marrow. And only about 30 to 40% of the population actually appears to have a cure. So, where are we at? Current thrust in treatment is directed almost exclusively, as far as I can tell, at eradicating B cells. As I said, I think that the research needs to go into not only that, we need to develop better ways of eradicating B cells if we're going to succeed at curing CLL, but we also need to develop ways of targeting potential mutated stem cells in order to completely eradicate the source of mutated B cells, CLL B lymphocytes. Okay, thank you. That's my thinking on the topic. I hope that what I'm doing by this is maybe stirring up a few people's ideas. Remember, this is my personal thinking based on my reading of the literature. It is not something that I have read anyplace, but at least it might be something to think about. If you liked this, click the thumbs up. If you didn't like it, click the thumbs down. Doesn't bother me at all. But if you do give me a dislike, I'd appreciate a comment to tell me why. And also, if you want to hear in advance when my new videos are coming out, go ahead and subscribe to my channel. Thank you very much.